Okay, so the previous class we have started the chapter of biotechnology and its application, and we wanted to know that what are the uses of the biotechnology which we have studied in the previous chapter. As we know that the biotechnology itself is a very cumbersome process, then what is the need of those things to do? Okay, so in the various fields, like in the field of agriculture, in the field of human health, and in other several other applications we are using biotechnology. So the introduction I already given. Today we shall study the further of that thing. So yeah, let me share the book. Okay, so we were studying about the use of biotechnology in agriculture, in the science of agriculture. So full screen, and यहाँ नाम मेरा जाए तो अच्छा हो जाए, so I can pin myself. So we have studied that the three areas of research in the biotechnology, that is to how to produce the recombinant organism, and then second to providing the optimum conditions, and third to that is filter and stabilize and test the product, and then its marketing. Then we are studying about the agriculture. The three ways agriculture are done. The one is agrochemical based agriculture, which we are doing nowadays, and the second one was the same uh, organic agriculture, which was our ancestors used to practice. They used to be dependent on the manure, and they used to be dependent on the what do you call, the normal method of pest control. They never used any sort of harsh chemical which used to cause direct damage to the environment by destroying the insects and other useful insects, and by destroying the soil microbes and all. So that is why organic agriculture was much better than the agrochemical based agriculture. But the problem was that in the organic agriculture was that its productivity always used to be limited. Now we got the third scope, that is the genetically engineered crop based agriculture. What is genetically engineered crop based agriculture means? That is, in this practice we will be using the high yielding variety which are genetically produced. And in the previous chapter, we already summarized that what ways genetic engineering is better than the simple process of hybridization. And whereas in the hybridization, we cannot control the entry of the undesired characters, but in the genetic engineering, we have an ability to control, and we can transfer only and only the gene of desired character and get that particular type of expression. And secondly, we also studied that the desired gene can not only be taken from only that species member only, but we can even get the gene from some other genus, other species of the same genus or other class or other order, or even from the different phylum or kingdom. Like that, we can get some of the gene from human being and put it into the bacteria and get that type of product. Because for the DNA level, it is the DNA. It is not that species or the kingdom or the order or the phylum or any other category which they recognize. DNA recognized as a DNA only, and the expression will be done. So that provides us the better scope. The scope is like we have to think what we want to get. Okay, from the one type of plant, whereas today we are getting only a kind of fruit or a kind of vegetable or only one kind of thing like the seed or grain or spice or whatever thing. If we insert some other characters like like from a plant of wheat, if we insert the jute fibers character from the jute fiber, tomorrow it may be possible that our wheat will also have this the this xylem fiber similar to that of the jute fiber. So along with the wheat, we can also get the, so the scopes are unlimited. Scopes are unlimited. We have to think in the right way, that right direction, and we have to carry the experiments in a better proficiency, and then have all, like, the problem only is that there may be chances of some wrong expression. So that has to be controlled. That has to be seen. Otherwise, the scopes in the biotechnology are unlimited. Okay. So now, see, now if I read from the Green Revolution succeeded tripling the food supply. No, it was not enough to feed the growing human population. Increased yield have partly been due to the use of improved crop varieties. So how we got the improved crop variety? Then we got by hybridization, and hybridization between the same species, or at the most between the different genus or the same species, so different species of the same genus. So only that much is possible. Okay. So there was a little chance or little scope of improvement. Okay. But mainly due to the better management. But what productivity, what we enhance 3 times, 4 times, 7 times, whatever we are read in the chapter 9, that is all because of made possible because of the management practices. That is better irrigation, better use of fertilizers, better use of pesticide, and all these are not a good practice, because the use of pesticide, use of fertilizers, they will ultimately cause damage to the soil and the environment. Okay. And the water also, if we indiscriminately use water, naturally the availability of the water in the soil will go on decreasing. So these are actually management practice, not the improvement practice. Okay. So that is why we have to look for another method. So agrochemical based practices are often expensive and further increase in the yield. Existing variety are not possible using conventional breeding. If you go on using this type of breeding only, so a limit will come, a threshold will come, not more than that. Suppose from a hectare of land we can get 3 quintal of certain type of pulses, then that is the limit. You cannot make now this 3 into 3.5 or 4 or 4 to 5 and 5 and 6 and all. Okay, because that is the threshold limit, that is the maximum which you could get from that particular variety. Okay, so the scope are limited with the agrochemical limit. That organic farming had a very limited scope. Agrochemical are also having limited scope. So is there any alternative path for the understanding of genetics? Can we show that genetic can show so that the farmers may obtain the yield from their field? Yes. What? By genetic engineering we can insert only a desired gene. Again, so it has a number of benefits. Now let us summarize the what are the benefits we can get by the GMO plant, that is when genetically modified, when the foreign genes are inserted in any organism, it is called genetically modified organism, because we have changed the genetic makeup of that organism. So what are the scope, what are the benefits that we get? Okay, so that are summarized here. Made crop more tolerant to the abiotic stress like cold, winter, cold, drought, salt, and heat. Okay. Now the plants are more capable of resisting all these type of adverse conditions. Severe winters, they will be able to. You know that now the winters are going on, and several part of the north, in northern India, the snowfall are there. The snowfall can damage our crop. Again, because what happens, the water inside the xylem and phloem that freezes, and with freezes we know that the volume of the ice is more, so it cracks their xylem and phloem tissues, and then those cells, the water conduction affects, and then those cells dies. Again, so there have to be some sort of ability to resist that. So these plants, because we know several wild varieties are there, they can easily cope up with that type of things. Maybe because their xylem are strong, or maybe because there are some other type of mechanism. Okay. So those type of genes, if you are inserted into them, they will be able to resist severe cold. Same is with the drought, same is with the salt, that is saline water, and heat. Second, reduce reliance on the chemical pesticide, because we have inserted the gene from the biotic resistance genes from the wild variety, wild relatives. Wild relatives could easily succeed. You see, so many plants are growing here, so many plants are growing there. No one is putting any pesticide, but they can resist themselves and they can grow. Even in the lack of water, even when there are so many different type of attack of the insect and other type of pathogens. So these all are resistant variety. If their genes are taken and put into our crop plants, they will also become resistant. And in that case, we need not to depend on the artificial Pesticides, we'll just study about the Bt cotton and Bt variety of other organisms, this particular application. Next is help to reduce the post-harvest losses. There are several fruits and vegetables which have very short lifespan, um, like those tomatoes. Again, now we got tomatoes with the thick skin so that it can easily resist, or its shelf life is increased from 1 day to 1 week or so. Okay, so these are just because of the taking the gene from some of the other plants in which the shell is thick, and that is outer exoskeleton is not exoskeleton, that what you call, the outer layer is thick enough that prevent the loss of water, and it remains fresh for longer time, or it is thick enough so that even if you put 3, 4, 5 layers of those fruits, like when the tomatoes are carried, you know that carrots are there in which so many layers of tomatoes are there. So 8, 10 layers of tomatoes are in the top layer are applying constant pressure on the lower fruits, yet they do not crack or damage or burst. Okay? So that is because of the thickness of the cell. So that we have made by genetic engineering. Okay? Increase efficiency in the mineral uses by the plant. Okay, so it is also a very important thing, so that the soil will have uniform use of the different type of minerals, and enhance nutritional value. Like we want to increase the vitamin A content, so we have made this golden rice, a rice in which the creatine amount was more, so it appeared somewhat reddish in color. So that used to be a source of vitamin A. Okay, now in addition to these, the GM is also used to create the tailor-made plants. Now, tailor-made plants means it is not that the plant you will go to bring to the tailor and ask him to, yeah, yes, either tailor-made means as per our requirement, whatever is specific requirement. Now, when you go to get a particular dress stitched to a tailor, you may ask, you know, what type of collars you want, what type of buttons you want, what type of pocket you want, whether you want a flap or you don't want, whether you want a pocket or you don't want, whether you want the pocket on the left hand side or on the right hand side. All these you can decide. Similarly, you can decide a plant to have how much starch, how much vitamin, how much this thing. So just now that golden rice, we have enhanced the quantity of vitamin A. That was a tailor-made character. Similarly, the industrial purpose, we use several type of things, like the guar is used to make gum. So the gummy substance, it is a type of starch. So increasing the content of the starch, or for the fuel, and for the pharmaceuticals, when we want a particular type of oil, like eucalyptus oil is used for the various type of medicine. So we want that the amount of eucalyptus oil should be more. So we can use such type of variety in which the amount of these type of things can be, or product can be more. Okay, just a minute. Okay, so these are the advantages of biotechnology over the simple experiments. Okay, the simple agrochemical and organic based agriculture. Now, one of the example here given is that is the reduce on the dependence of pesticide. The very first variety of pest resistant variety was introduced in India was Bt cotton, where the Bt is standing for Bacillus thuringiensis. Bacillus thuringiensis is a type of bacteria, and when this bacteria used to be attacked by some insects, those insects used to die. It has been noticed by some of the scientists. That is, whenever one certain kind of insect used to eat these bacteria, those insects used to die. On searching, they came to know that there is a kind of protein which is found in them, which is in the form of inactive protein, but once it goes into the intestine of these bacteria, these insects, they become active and they create perforation in their intestine, and because of those, they used to die. So then scientists search for the gene for that particular protein. We simply name it as a protoxin. So that particular protoxin gene was identified, isolated. Say, for example, this is the one of the chromosome from the bacteria, in which the gene for this protoxin is found from here to here. This has been cut, cultured, and then it is inserted into the embryo of any seed, any plant seed. Okay. So once it is inserted into the chromosome of embryo of any of the seed, so whenever the seeds will germinate, it will produce, increase the number of cells. All the cells will have this gene from the insect, bacteria, Bacillus thuringiensis, and all the cells will be able to produce that protoxin. Now, there are two things which we have to be very clearly understand. That first of all, the protoxin, the toxin is found in the form of inactive form, that is protoxin. Okay, so it will become active only and only when they come in contact with some basic medium, alkaline medium. In human being who are the consumer of most of these vegetables or fruits or whatever type of plants in which we are putting these genes, it will not affect us, because in our intestine, our stomach, well as a digestive tract, mouth does not have any acid or base secreted here. Even in the esophagus, there is nothing secreted. The first chemical, that is the medium, is secreted in the stomach, and which is acidic. And in the acidic medium, proteins will be denatured, but the protoxin will not take change into toxin. So it will not affect us. It will not affect human being, but it will affect only those insects in which in their alimentary canal, in place of acid, bases are secreted first. Okay, so there are some category of such insects. Their names are given here. That is, these are the Lepidopterans, then Coleopterans and Diopterans. Only these category of insects, when eats these plants, they will be secreting some bases, and when this protoxin comes in contact with those bases, it will become into active toxin and it will kill them. So we have to see that in what plant the Bacillus thuringiensis gene can be introduced. We have to see whether the pests which are attacking on them are one of these category. Are they of Lepidopterans or Coleopteran or Diopteran types? If they are not belonging to any of these three, then it is no use. Okay? Because only these are the organisms in which the basic environment is created in their digestive tract first. So the protoxin will become active toxin, and that will kill them. For others, it will not affect. So it is a narrow spectrum. We know that which type of organism it will cause damage to and which kind of organism it will not cause damage to. So we need not to worry. Okay, so that is very important thing. Okay, now specific BT toxin genes were isolated from Bacillus thuringiensis, incorporated into the genes of several crop plants such as cotton. The choice of the genes depend on the crop that are targeted pest. Again, one of these category which I just explained you. The most of the BT toxins are insect group specific. The toxin is coded by the gene named as CRY. Again, the CRY is actually stand for crystal. So the gene, हमने gene का नाम दे दिया CRY. Now where is the location, which place they are obtained from? That is given as this type of name, that is CRY1AC or CRY2AB or CRY1AB. These are the various type of proteins which we obtain, and they will be controlling the different type of crop. So CRY1AC and CRY2AB will be controlling cotton bollworm, and CRY1AB will be controlling corn borers. So you can see here the diagram of corn, not corn, this is of cotton, and cotton will this is the healthy cotton here. This one is the healthy cotton, and this is the cotton which is eaten away by the bollworm. Here you can see the bollworm. So the bollworm eats away the cotton, and the productivity is greatly reduced. So now what we did, we removed the bacillus gene, Bt gene, and that is the name of the gene is given there, like cry1ac or cry2ab. These genes are cut and then they are incorporated into the embryo of one of these plants. So all the cells will have this gene and all the cells will be producing protoxin. So whenever such a cell will be attacked by one of these kind of bacteria, so those bacteria, one of these kind of insect, those insects will die and our plant will be unaffected from the what you call these pest bollworm. So the farmer has tremendous result. Why? First of all, their dependence on the chemical is reduced. In one crop, at least 3 application of pesticide required when they were depending on the pesticide. 3 application of pesticide. Suppose someone has got a 2-hectare land. So first of all, you will have to invest a lot of pesticide, buy a lot of pesticide, make solution. Then you have to put a lot of labor in order to spray that. And that is one thing. It was economically expensive. It is labor oriented, and the impact onto the other organism, the good organism and the bad organism, are equally same, because pesticide cannot differentiate between good and bad. So it used to be killing non-judiciously the useful insects as well as the bad insects. And then other pesticide which used to fall on land, or the pesticide which falls onto their leaves, and such leaves are consumed by some of the cattle or some other organisms, they used to also be affected. When the pesticide used to fall onto the soil, then the soil microbes used to be affected. When it used to be taken away by the nearby to the nearby water bodies, the water used to be get poisoned and it used to enter into the food chain and all. So the loss, the effect of the loss was greatly reduced by use of Bt oil. So first it was used for the cotton plant. Now today we know there are number of variety of organisms for which the Bt gene is used, like Bt cotton is there, Bt corn is there, Bt rice is there, Bt tomato is there, Bt potato is there, and Bt soybean. Again, etc. is written. That means many more are there. So if farmer use these variety, of course the seed will be little expensive, but the benefit which they are getting are far more than the actual input, because they will be creating an IPM, that is, whatever the IPM, full form of the IPM, integrated pest management. That is, we have to maintain the number of the pests to a level where they may not cause destruction, but they will also be there as a part of food chain. Okay, so they will be maintaining food chain, they will be maintaining the species also, not allowing them to completely eradicate from the same universe, and also protecting the soil and water and other type of system. So the use of Bt was very good example. Okay, so the question which comes in this, you may ask, what is Bt, what are the why it is not harmful to the human, or why it is harmful only to these category, or what type of insects which it will be harmful for, and what ways the use of pesticide, this Bt plant had a good to the environment or to the other organisms and all. So that all you can write. Then you can also write that what is the role of acid or base pH, and when these type of proteins are consumed by insect, what actually happens. So we have to see that these inactive proteins are changed into the inactive protoxins are changed into inactive protein जो है ना, that will change into active protein या toxin, and that will cause the pores in their digestive tract, and that is how they die. So I think that is enough for today. नया टॉपिक शुरू करने लायक टाइम नहीं है, so we should give it a rest. Okay, you can ask your problem if you got any.